We are very excited to have with us Dr. Adrián Villalba Felipe. He's going to be presenting his talk, A 3D Atlas of Human Developing Pancreas to Explore Progenitor Proliferation and Differentiation. This paper just recently came out, and it was really well received. It's a beautiful paper. I encourage everyone to take a look at it if you can, and we'll put the link to the paper in the chat in just a moment. But I wanted to introduce Dr. Villalba Philippe himself. He's at INSERM in France, in Paris, and a short bio about him. He's a vocational scientist and intellectually eager, strongly interested in cell biology processes, which can be focused in diabetes research. Biomedicine has become very collaborative since the last decade, and many different scientific disciplines such as immunology, surgery, stem cells, or medicinal chemistry try to converge in order to give satisfactory outcomes to the improvement of life with diabetes. I find it very exciting to work in a multidisciplinary environment. It lets me learn steadily and meet people with shared interests. The fight of diabetes at this point seems to be difficult, but the possibility to translate results from the bench to bedside is the greatest satisfaction I could aim for. And these facts sort of made me fall in love with this topic, type 1 diabetes. And so without further ado, thank you so much for joining us, and we really can't wait to see more from your work.
Thank you very much, Monica, and thank you very much to all the team of the Super Science. I think this kind of series are extremely interesting for all the community that we used to work sometimes quite isolated ones from each other in diabetes, and I think it's a good opportunity to join forces in order to fight against diabetes. So thank you very much for everyone coming today. I want to begin first. I'm going to present the results of our last paper published in Diabetologia, which is the basis or a very preliminary 3D atlas of the human developing pancreas that we think it's nice to chart to explore progenitor proliferation and differentiation. This work has been performed between the close collaboration between two groups, both of them based in Paris. The one of Raphael Schaffman, who is a long-standing researcher in the field of diabetes, and the one from Alain Total, who is trying to make these interesting 3D atlases of human developmental cell atlas. Before we begin, I want to explain a little bit my background and my interest in diabetes research. So I earned a PhD in immunology almost 4 years ago in 2020 by the Autonomous University of Barcelona. And there I studied the development of a therapy to treat type 1 diabetes in mouse models, in mouse treated with streptozotocin, and we repurposed liraglutide, which is an agonist of GLP-1 receptor. And my main work was published in Frontier in Endocrinology, in which we show that after streptozotocin depletion of beta cells with an agonist of GLP-1 receptor, we were able to induce alpha to beta cell transdifferentiation and also neogenesis of beta cells from the ductal part. Since 2020, almost 4 years ago, I am postdoc at the group of Raphael Schaffman, who is quite known in the field because he was the first to develop human beta cell lines, the BH, BH2 till BH5 beta cell lines, using this technology or part of the biological sources that I'm going to explain to you today, which is to take human fetal pancreas transduced with a rat insulin promoter expressing human third SV40 some genes in order to increase the immortalization of beta cells. This human fetal pancreas was then grafted in skid mice, and they were able to obtain at the end fetal beta cells able to proliferate in vitro and to secrete certain amounts of insulin in response to glucose. Finally, I am LabX Revive Fellowship since a couple of years ago, and without any other ado, I'm going to go to our project. So I also have type 1 diabetes. For that reason, all my entire research life has been focused on diabetes or in pancreas development. I am part, or our group, Rafael Sharfon's group, is part of the islet consortium, which is an islet project in which there are many partners in Europe. Is guided, or the main director of this project is Henrik Sem in the Helmholtz Center in Munich, and there are many great scientists in this group, François Carlotti, Elko de Koning, Heiko Lickert, among many other teams. And our main goal is to generate the first cellular therapy for treating type 1 diabetes in Europe. So this is not something new at all. We know that in the States, in America, and even in Asia, since many years ago, we have the technology to produce stem cell derived beta cells that once they are transplanted or grafted into diabetic mice, they are able to recover not only the basal glycemia, but also the capacity to respond to glucose stimuli. Since some years ago, Vertex has prompted one and then second trial. The first trial was suspended for some time last year or at the beginning of 2024, but since April 27th, the first trial of Vertex has been resumed, which I think is great news for the patients with type 1 diabetes. And these trials consist of stem cell derived beta cells, non-encapsulated or encapsulated, in order to treat patients with type 1 diabetes. So I guess I'm not going to enter into the details of type 1 diabetes, because I guess that after many programs here in the sugar science, everybody should be more or less familiar with the disease. But I
I hope
want
so.
to not so okay.
No,
So
no, I said, I hope so.
okay. So, but I know that most of us are familiar with diabetes, but maybe not with pancreas development. So I'm going to have 1-2 slides of brief introduction. Everybody knows that islets of Langerhans are clusters of monohormonal producing releasing cells that are clustered in the pancreas, and we know that these cells are more or less 1 or 2% of the pancreatic cell mass. The vast majority of the pancreas is made of acinus, which are exocrine tissue secreting digestive enzymes to the ductal tree that will be released to the duodenum to participate in the digestive function, while on the endocrine side, which are the islets of Langerhans, these cells will release the hormones through capillaries or through blood vessels to the circulation, and will act on different cells in the body. How this incredible organ, which is a mixed gland with an exocrine and an endocrine function can develop? So, well, first of all, most of the data we have comes from murine pancreatic development. That means that we don't have too much information on human tissues, because it's quite difficult to obtain these tissues. They are very rare samples, and we guess that something that happens in mice always parallels to human, but it might not be the case. For that reason, I will explain later. I think our atlas is interesting. First of all, pancreas emerges during development as two independent buds. The first bud is the ventral, and the dorsal one, which is the larger. Then there is some kind of elongation of these both buds, and there is rotation of the full ventral pancreas and the gallbladder. Then both ventral buds will fuse and will converge into the single pancreatic primordium that we are used to see in adults. This is what happens at the macroscopic level. If we go to the microscopic level, I really love this figure, because here we have the comparison between mouse and human. And most of the boxes for human, especially in the earliest stages, from the day 23rd to the 6th week of development, even the 8th week, are completely empty, because we don't have too much information on this. And I think we could provide in our recent paper some clues. But mostly in mice, what happens is, after this rotation, both buds fuse and grow together. And in parallel, there is differentiation of the different cell types. Here we have pancreatic multipotent progenitors that will give rise to the three different cell types. That means acinar cells, ductal cells, and endocrine cells. Intriguingly, ductal and endocrine cells have a common progenitor, which are trunk cells, and in the tips we have this kind of tip progenitors, which will give rise to acinar cells. So we wanted, or we aimed to fill the previous gap that we have seen in the development of, in the early stages on the development of the human fetal pancreas. So we recently published this article in Diabetologia, and we claim that there is more or less quite information about the rodent pancreas development, which is well documented, but the underlying mechanisms for human pancreas formation are less understood. It is true that with single cell technologies, we are able to identify a lot of transcriptomic information and gene expression levels at different stages of the human fetal pancreas, but they lack of the 3D or the spatial information that we have in 3D or in a topographical manner. So the information that we consider quite interesting, like cell contacts or cell type rearrangements during cell fate organization, during the differentiation from pancreatic multiple progenitors, are not captured by single cell technologies. So here we benefit from a technique in which we can image the full pancreas in order to ask questions related to the proliferation and differentiation of the first pancreatic progenitors and see them in a full manner in the whole organ. So I'm going to present now the results, and I want to begin with this quote from Sidney Brenner, who was Nobel Prize of Medicine in 2001 for seminal work on cell death in nematodes. And he claims that the progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. So here we use a new technique or a pipeline, which is the combination of quite new techniques in order to get new insights and from these insights build new ideas. So it's common to build now 3D atlases. We have access to embryos, which is the developmental stage till the 8th week of development, and to fetuses during the first trimester of pregnancy, which lasts here in France till 12th, 13th, or even 14th week after conception. And all of them come from voluntary abortion. So they are very rare samples. It's quite difficult to obtain them and to have the ethical permissions to access these samples and to perform experiments with them. So we perform first a biobank of these embryos and fetuses, and then we perform a long protocol which takes almost 1.5 months per sample in order to do in toto staining, which means to stain the full organ. Then we perform tissue clearing, which means the delimitation of the tissues. That means to remove the lipids in order to have a refractory index between the sample and the microscope quite adapted that will align, so we can have the theoretical maximal resolution. And then we perform 3D imaging. I'm trying to see, okay, if this works. This is a full human embryo at around post-conceptional week 8, in which I have manually highlighted the pancreas in purple. So here, this is light sheet fluorescence microscopy. What is interesting is that in confocal, we are able to send the laser beams in one direction and recover the excited light in the same direction. So the angle is 0 degrees. Due to that, we are able to have a confocal volume, and we can repeat this in every position in a single slide. Which issues are related to confocal imaging? It is true that we can have 3D images, but first, we can have them because they are in a single slide. So the thickness will not range the full specimen or even the full pancreas in the case of human embryos or even human fetuses. And the second issue is that to get different volumes allows us to stack and to concatenate them and to have this kind of Z stack or 3D spatial depth, but it takes a lot of time. So if we want to have this kind of resolution in an embryo, even if we section this embryo slice by slice, it will take years. In light sheet microscopy, we are able to circumvent this issue by sending the lasers at 90 degrees to the direction in which we recover the light. So we keep one 2D sheet illuminated, and then we are able to stack the different light sheets and to make them into a full 3D object like this one uh, that I come here to show you. And these techniques allow us to ask interesting questions that can only be solved in the full specimen. Just a disclaimer before we begin. DOX9 is a well-known marker of ductal cells. PDX1 is a well-known marker of beta cells, and NKX6.1 is another transcription factor restricted only to the endocrine portion of the adult pancreas. But here in the human fetal pancreas, all three coexist. Here you can see the staining of a human fetal pancreas. Here we have the head of the pancreas. Here we have the belly. As you can notice, the shape is not really similar to an adult pancreas. And these three markers coexist in the different epithelial cells. You can also realize that there is a huge difference between the shape of the epithelium in 2D and in 3D. Here, what we have is an epithelium, if we look at the highest magnification, an epithelium with a lumen in which at some point, we are not sure when, um, acinar cells will secrete dairy content, and they are surrounded by mesenchymal cells, which are less common in the adult human pancreas in healthy conditions. And all of these epithelium tend to fold one between the other in order to recreate this structure. First, what we did in the paper was to locate, here I show you at 7th, not 8th, post-conceptional weeks, these specimens stained with SOX9. Here we can show that at this stage, the pancreas is already located in the left upper part of the abdomen, like the adult one. And here again to show in 2D how is the shape of this epithelium in a virtual slice of this lens sheet area next to other SOX9 positive epitheliums like the one of the gut or the one of the kidney. Next, in mice, we know that there is this bud rotation. So we have the ventral bud and the dorsal one, and they rotate in order to fuse together. Then we will have here the main duct of the pancreas, which comes from the fusion of the dorsal one with the ventral one, and will drain to the duodenum, which is common, will join the common bile duct that drains to the gallbladder. And on the other side, we have the Santorini's duct or the Santorini's Santorini's duct actually that comes only from the ventral one. This we know that happens in mice. We guess this happened already in human, but we were unsure about the timing. And almost by surprise, we were able to catch the very first moment in which here at PC week 5.7, which is almost 1 month and 1 week after conception. So it's a really rare sample, a very early abortion. We were able to see here that the head of the pancreas was bilobulate. Here we have the ventral bud and the dorsal bud. And 1 week after, this is a different specimen, at PC week 6.4, we are able to see the fusion in the pancreas division of the head of the ventral bud and the dorsal bud. This is interesting for matters of differentiation that I will explain in one of those slides later. But I don't know if we can see if I can click here, maybe I will remove this in order to put this video. So both, because in the picture sometimes it's difficult to see, these two buds are already disconnected. There is no physical contact between both of them. So it seems that the emergence of these two buds might occur much before PC week 5, which is really early in order than what was thought before. And these buds are already about to fuse. So even if we don't have access to really, really early specimens, we might hypothesize that emergence of pancreatic buds happen at around 3.5, even 4 PC week, which is much before it was not before. And here we already see them already rotated and about to fuse. I go to the next slide, and I also use the laser. Here we show just a macroscopic view from PC week 5 to PC week 11, which was the first window or time window that we had to access these different pancreas. Now, with new laws in France related to abortion, we are able to obtain samples at PC week 12, 13, and some at 14 as well. Here you can see that at PC week 5, we have both, again, the ventral and the dorsal bud. Dorsal bud is much larger than the dorsal one, and this is something concerned across the species. We have seen that in primates and also in mice and any kind of rodents. Then the pancreas grows as expected by increasing the length, which is this axis, and the diameter, which is this axis. And when we compare the ratio between both of them, we see that this is more or less common. So here we found that the pancreatic morphogenesis displays an isometric growth. That means it grows in both directions, in both the X and the Y axis. The next question that we are trying to answer, and maybe could be able to give rise to another story, is to see if the pancreas that grows isometrically at this point grows by layers. That means only peripheral cells can add an extra layer, or they can glow like a balloon model, which is the case of the lung, for instance. Then we ask, okay, we are able to see the full pancreas. We are able to ask questions that cannot be asked in 2D because it's quite difficult to have all the pancreas to slice the full pancreas in a very exact manner and to image all of them, because here, if we want to have 4 micrometer thickness slices and to slice all the pancreas, we can have 50 or 60 slices. So it's quite difficult to go to the confocal and reconstruct that. But here with this technique, we ask this next question. When do the first beta cells appear? So we stain for SOX9 as a pan-epithelial marker with insulin, which appears here in white. Here we have the ventral bud, the dorsal bud, and we are able to see the first insulin-positive clusters here that are appearing at around PC week 5.7. Much before, almost 2 weeks before it was expected. There is a very nice paper already published in Diabetology in 2012 by the team of Neil Hanley, in which they show that there is NGN3 positive staining in human fetal pancreas at around PC week 7. So they thought that, with slices is the true conclusion, I think, that first insulin positive cells might appear at around PC week 7. Now we know that they appear at around PC week 5, which is 2 months before and might have implications for the development of stem cell derived beta cell protocols. And it could give us an idea that we, if nature can, maybe in vitro, we are able to shorten a little bit this process of differentiation.
Adrian, can I ask a question?
Sure.
Between the last SOX9 insulin marker on the far right and the tail, it's pretty dark. Is that just because it's out of the plane of focus? Or
Yes, this is,
what's going on there?
No, this is because we are taking light sheet areas from here 2 here, and in this case,
Yeah,
it was not in the, we adjusted to have the focus more or less in the middle of the pancreas, and here it was a little bit overexposed, and here it was not exposed at all.
I see.
If we pay attention to the distribution of the insulin positive clusters, we can see that they are restricted only to the dorsal bud and not to the ventral one. This is consistent with previous findings in both mice and zebrafish. I am thinking of similar work led by Pedro Herrera in the University of Geneva. And this prompts us to ask if there is a neogenic niche related only to the dorsal bud. Not only an intrinsic one, but also another one related to the spatial location of this bud. And because we don't have access to earliest samples, we cannot see if this differentiation comes before the rotation of both the ventral and the dorsal bud.
Here's a great question in the chat from Lena Sweet. Are the first wave of beta cells polyhormonal?
This is a question that we are trying to unravel right now. Here in this paper, we only stain for insulin, but this is preliminary data. I found in the last months that at PC week 6, at least, we have hormonal insulin and somatostatin positive cells, which is something that I have not seen before in humans, even if others have looked at this. The main issue is that not all the cells during the first transition wave of differentiation are insulin and somatostatin positive, RB hormonal. So I suspect that some people looking at slides could see them only monohormonal and claim that the first endocrine cells are monohormonal. But if we look in 3D, we find rare double positive cells, at least for insulin and somatostatin. So in this case, I don't have the staining in this specific sample, but in other samples, we have seen that there are some bihormonal cells for insulin and somatostatin. This is also shown by papers by, there is one paper in endocrinology reviews by Jacob, which is quite interesting, showing also polyhormonal positive cells in mice at very early stages in confocal with high resolution.
Interesting.
So,
Here's another question. Have you tried
yeah.
staining for EEC cell markers like SLC18A1?
No, we have not tested for them. Actually, during all the paper, yeah, we try to claim that these cells that we call beta cells, in theory, are beta cells, but in practice, we call them insulin positive cells because we are not sure which other markers might express, and we did not look neither at other endocrine cells at this stage, neither for enterochromaffin markers, neither in these stages, neither in other cell types.
Okay, great.
So now we have answered the first question. When do first insulin positive cells appear? And we know that if we go weeks before it was expected, we still find insulin positive cells. What we also found, and this was interesting because it did not come from a direct question, is that we were able to find extrapancreatic insulin positive cells in the gut. We detected insulin positive cells in the fetal gut in 40% of the specimens. Here I have reconstructed, this is the common bile duct, the Santorin is that. Here we have the pancreatic epithelium, and this is the gut. And clearly we can see insulin positive cells in the gut. But previous data already claimed the existence of insulin positive cells in the gut. This is an interesting paper from the group of Itzkovitz, published in Nature Medicine in 2021, showing that there are some insulin positive cells which are most likely to be L cells of the gut, both expressing insulin at RNA level here in purple, and also at protein level in green. And we were able to confirm this that was from experiments in single cell RNA-seq. Next. Okay. I think I have to. Okay, yeah, this is 1 second. Okay, this is just to show that it's not an artifact of the 2D picture, but if we look at 3D, we have embedded into the gut epithelium these different insulin positive clusters. Then the next question was evident. We know when do the first beta cells appear. We know that they are located somehow, or some of them outside the pancreas, but in the pancreas, where are they located? First of all, we counted the different insulin positive clusters across development in our time window between 5 and 12 PC weeks. We detected that in this period between 5 and 7.5 PC weeks, even previous authors have claimed that in 2D there were no insulin positive cells. In 3D, we were able to see some cells, and this is the density. But as you have seen, the insulin positive clusters contain no nuclear marker because SOX9 is present in the pancreatic epithelium but not in beta cells, because during the adulthood it will be restricted only to SOX9 ductal tree. So we performed the same staining, but in 2D we counted each insulin positive cell per cluster, and we found that some of the clusters that we are able to detect are uh, monobeta cells. Some of them contain 2 or up to 4, 5 insulin positive cells per cluster. So this confirms that we have single cell resolution in 3D. And
So
when we
Adrian, are you guys hypothesizing that the beta cells form the initial islet and then other cells begin to populate the islet, or what?
this is current experiments that we are trying to do. We are measuring the differentiation time of alpha and delta cells. It's
Mhm.
true that in mice, glucagon positive cells are the very first to appear, even before than beta cells, and are also the first to populate islets. But
Mhm.
our preliminary data, we don't have much samples now, it's still preliminary. What we have seen is that insulin and somatostatin positive cells are the first ones to pop up in the human fetal pancreas, that glucagon positive cells appear much later
Mm.
than insulin and somatostatin, and that conversely to mice, beta cells are the ones um, being the first to populate the islets and are located in the very core of the islet, which is somehow interesting because in in mice, it's quite clear when we look in adult pancreas that insulin positive cells are in the center and then they are surrounded by alpha and delta cells.
Mhm.
But in human, they look more or less stochastically distributed. It's not random, but a huge proportion of alpha and delta cells are located intermingled with beta cells in the core of the islet. And we have plenty of beta cells in the mantle of the islet.
Yeah,
But in the
the murine in the human morphology is really different.
Exactly. But when we look in the human fetus, what we see is an islet that looks really similar to the adult murine islet.
Hmm.
So taking this data into account, now we are trying to make some hypothesis or to claim something about islet fission or fusion in order to unroll a little bit how islets form during human ontogeny, which might be different to the fission model that we have in mice.
Yeah, that'll be interesting.
Here, what we did was to compare with mouse, and basically what we see is that, as expected, in stages that are similar to human, we are not able to detect any insulin positive cell, while later, even if we still have somehow these two kind of ventral and dorsal buds, we are yet able to detect the first insulin positive cells popping up here in, in mouse. But this does not answer our initial question, where do beta cells appear? So to do that, we perform stainings at different stages of insulin and SOX9. Here we have the transversal view and the sagittal view. Apparently, it seems that the distribution of insulin positive clusters preferentially located in the inner part of the pancreas. So what we did was to section in the sagittal view the full pancreas to measure the distribution of insulin positive cells to the centroid of the pancreas, which would be the mathematical theoretical axis, because this is not a cylinder, so we cannot have a center, but we can have a centroid, which will be a center-like structure, and we plot the distance for each sample of these insulin positive clusters from the periphery to the center. So here in the 0%, we have the center, which will be this dot, and at the 100%, we have the periphery, which will be this dot. And we can see that in the first two quartiles, we have the vast majority of these cells, and this will be the mathematical demonstration of what we see by eyes, that insulin positive cells preferentially locates in the inner part of the pancreas. So we know when the first beta cells appear, we know where are they located. We are tracking here the differentiation of beta cells. I'm going to show you again another specimen. Here we have the pancreatic epithelium stained with SOX9, and at some point we are going to have an overlay of the insulin positive clusters. And if we turn it around, we can clearly see that in the tips, in the periphery of the pancreas, there is a huge lack of insulin positive cells. So in the inner part of the pancreas, we have a neogenic niche of differentiation for this kind of cells, at least. We don't know if it's shared with other endocrine cells yet, but we have clues to claim that it's similar to glucagon and to somatostatin positive cells in the human fetal pancreas. But which factors can be related to differentiation? Because if insulin positive cells appear in the center, this means that there is some factors inducing the differentiation of pancreatic progenitors in these very specific areas. And we thought about vascularization. But we found that there is no vascularization in the insulin positive cells. We look at CD34 blood vessels or even capillaries. The pancreas contains some blood vessels, but there is no blood vessel contacting directly the insulin positive compartment. And then if we look through muscle actin, if we look directly to arteries, we can see that there is no main artery draining the pancreas at this stage, which is something more or less known. So, intriguingly, these cells are not vascularized at this stage, and it's not that they are not related to the differentiation of the first insulin positive cells, but it also prompts us to claim that if first insulin positive cells are not vascularized, it is also logic to think that these cells are not secreting insulin because they don't have any means to do that. Then we thought, okay, now we have tracked the time and the space of differentiation, at least for insulin positive cells. And we have seen that the human fetal pancreas and the embryonic one as well, the human developing pancreas, is somehow regionalized in the differentiation of insulin positive cells. And differentiation is in some tissues a process that is quite opposite to proliferation. Should we expect a different profile of proliferation in the human fetal pancreas? And this would be quite interesting for our stem cell derived beta cell models if we want to increase the pool of progenitors to have a huge or a higher yield of stem cell beta cell derived material at the end. And we measured the differentiation of progenitors, taking into account that they are PDX1 positive by day 67, and we saw that between PC week 7 and 11, most of these progenitors are proliferating at around between 20–30% of them are proliferating. But if we look at insulin positive cells, it's quite strange to find insulin positive cells that are Ki67 positive plus. Here I show the optical magnification of 1 that we found, but overall there is between 0.4 and 1.5 of insulin positive cells that are proliferating. So here we come from insulin positive cells that appear in the inner part of the pancreas and are not proliferating. And then we have a lot of pancreatic proliferating progenitors, which are located in the full pancreas, but we don't know where are they proliferating. So if beta cells are differentiated in the center, where are located these proliferating progenitors? And this is the next question we ask. But first of all, we wanted to be sure if kinase67 was a good marker to claim that during differentiation in the human fetal pancreas, there is no more proliferation. And we tested for 40 or 35 different proliferation markers, BCNA, again, kinase67, KIV, aurora kinase B, etc. And we sorted through different membrane markers, 3 different stages of differentiation in the human fetal pancreas. Here in red, we have pancreatic multipotent progenitors, that is, progenitors that will differentiate into acinar, ductal, and endocrine cells. Here we have endocrine progenitors, that means they will give rise to alpha, beta, delta, gamma, and epsilon monohormonal cells at some point. And in blue, we have only endocrine cells. Here we have a pool of alpha, beta, delta, and the rest of monohormonal cells. And we saw that it's not only Ki67, but globally, these 35, 40 proliferation markers that hugely decrease from an undifferentiated stage till a more differentiated stage. So it's true that insulin positive cells do not proliferate in the human fetal pancreas because there is a huge debate about that. And then we want to ask the following question. If only progenitors are proliferating in the human fetal pancreas, where are they located? And to do that, we take advantage of EDU, which is a small molecule that can be intercalated during the S phase into the DNA strands. And we were able to pierce our tissues with EDU for 4 hours and then to reveal them. And here, if we look at the inner part of the pancreas, the progenitors, some of them are positive for EDU, but the vast majority are negative. And on the opposite, if we go to the periphery, we can see that in these ductal tree structures that contain mostly PDX1 positive progenitors, some of them are proliferating. But if we take into account the vast majority of the progenitors, the ones that are proliferating are preferentially located in the outside part, in the periphery of the pancreas. Here we have the mesenchyme, which is also proliferating, the peripheral mesenchyme, and the intrapancreatic mesenchyme, which is not proliferating at all, consistent with our hypothesis that only the periphery of the pancreas is able to proliferate in this manner. But again, maybe our eyes were wrong, and we wanted to mathematically demonstrate that. So we benefit from a mathematical tool, which is a function, the reliability function, which is used in spatial distributions. Here we can plot from one progenitor to another the distribution of the nearest progenitor. And if the distribution is random, like this one, we will have a profile similar to the random distribution, which is the red one. The observation should more or less overlap with the random distribution. If we have aggregates, we should have an observation increased or located over the random distribution. And if they are clustering in a regular manner, the observation should be below the random distribution. We took 3 different specimens at different stages at PC week 7, 9, and 11. And in 3 cases, all the observations were above the random distribution, confirming that pancreatic proliferating progenitors not only are located in the periphery, but are also clustering in aggregates, as we can see here in this picture. Then we ask this question, where are located acinar cells? Because we know that endocrine differentiated cells are in the inner part of the organ. We know that proliferating progenitors are in the periphery. Then acinar cells, where are they located? We see by Ki67 and the staining of carboxypeptidase A1 that some of these cells can proliferate, especially those that are in the periphery, if we compare with the ones in the inner part. But if we pay attention to that, not in all periphery, but in some parts of the periphery, we have acinar cells. So it seems that here mostly we uncover the tissue micro-regionalization of the human fetal pancreas. In the inner part, we have a niche of differentiation. In the outer part, in which cells, pancreatic progenitors, differentiate into endocrine progenitors and insulin-positive cells, but do not proliferate. And in the outer part of the pancreas, we have pancreatic progenitors that are proliferating and at the same time differentiating into acinar positive cells. This was quite descriptive, and we thought, okay, we have a very nice technique, which is in toto staining, tissue clearing, and 3D imaging. But can we switch to a functional model? Can we do more? Can we modulate proliferation? Because definitely, if we can modulate proliferation, we can incorporate this modulator into a protocol to obtain stem cell-derived beta cells, and this would increase the yield of beta cells simply by expanding the pool of progenitors. And we designed an extron culture system in which we have here a normal regular medium. We keep them with a filter, and this filter, which is a floating disk, we are able to keep here the human fetal pancreas. So the human fetal pancreas is in an interface between the air and the medium. This system allows us to maintain up to 7 days the pancreatic progenitors in proliferation, but not in differentiation. As we can see here, they are able, they still maintain the capacity to proliferate. They are somehow still kind of 67 positive. But when we look at pancreatic multipotent markers like NKX 6.1, they are not able to differentiate at all. They are only able to proliferate. So it's a model that allows us to study only proliferation. Then we observed by previous transcriptomic data that PDGF receptors, which are receptors of platelet-derived growth factors, well-known mitogenic factors in other organs, are restricted only to the mesenchyme. They are not expressed in the epithelium like EpCAM, which is a putative epithelial marker, and they co-express together with sialin, which is CD248, which is a classical mesenchymal marker. If we perform staining for PDGF receptor alpha and beta, both genes, we can see that the protein is widely expressed in all the epithelium, both the intra and peripancreatic, but it's not expressed in the epithelium, only in the mesenchymal fraction. So we raised the hypothesis if we could use PDGF isoforms in order to increase the proliferation of the mesenchyme, and this would increase the proliferation of the epithelium. And this is what we saw in control conditions in our tissue culture. We set up a culture by using noggin, nicotinamide, and forskolin in order to reduce the proliferation of the progenitors from 20 to 5%. And then if we fuse them with PDGF receptors for 3 or 7 days, we are able to increase threefold. We began by 20%, we decrease to 5%, and if we get alpha, we are able to go again till 17% of proliferation, which is threefold enough. So I will quickly go to the conclusions. Here I presented the first 3D map of the human fetal pancreas that allows us to track that human beta cells appear at least 2 weeks earlier than expected, and that they are located in the inner part of the organ, in contrast to proliferation, which is restricted only to progenitors, and that happen in the periphery of the pancreas. So the main conclusion of this paper is that here we provide for the first time the regionalization of the cell fate in the human developing pancreas. And additionally, as an ad hoc experiment, we proved that we are able to modulate the proliferation in the human fetal pancreas by using PDGF alpha isoform to induce in an indirect manner through the mesenchyme the proliferation of pancreatic progenitors. Now the last slide, what is next? Now we want to improve this 3D map, and now we are performing the stainings with glucagon, with somatostatin, in order to ask questions related to how these cells delaminate from the ductal tree and cluster in these proto-islets. How islets are formed, where are islets formed, which endocrine cells appears the first, in which proportions, and so on. We are also performing stains with new markers in order to try to identify the earliest beta cells which are not yet expressing insulin, and to check if we can track the dynamics of differentiation across development with these markers. We are also staining with CPA1 positive markers, which is to delineate if we can find niches for the differentiation of acinar cells, because we know that they are in the periphery, but apparently they are stochastically distributed in the periphery. So we want to know if there are factors that can induce the differentiation of acinar cells in a very specific manner. And this hopefully would be the next story of our lab. We are currently performing the preliminary experiments in order to have the data. And I want to end this talk with this quote from Louis Pasteur saying that there are no such things as applied science, only applications of science. And we hope that our 3D atlas will be useful to improve protocols in order to obtain stem cell derived beta cells that hopefully will get at some moment into the clinic and will be a really valuable tool to fight type 1 diabetes. Thank you very much to everyone for coming.
Thank you, Adrian. That was some beautiful work with some really great outcomes. I'm opening it up to questions from the audience. People can either put their hand up or they can drop something in the chat. I had a quick, I'm looking across the audience for hands up. But I had a question for you regarding when you saw some early insulin secreting cells in the gut. When, you know, those, can you remind me when those turn on and when they turn off? I don't know if you followed up on when they turn off.
Do you mean the very first insulin positive cell?
Yeah, well, the ones that were actually, I thought you said they were secreting in the gut itself, not in the not in the precursor pancreas.
Yeah,
Yeah,
here,
there. Yeah, in the gut. And when do those
yeah,
turn off? Did they continue to stay secreting insulin in the gut after the
here,
pancreas is, you know, created? Or yeah, the ones that
yes.
are insulin
Well,
positive in the gut.
first, we don't know if these cells are secreting insulin, as well as we are not sure if the insulin positive cells in the pancreas are secreting insulin at this stage. In the
Right.
pancreas, we know that there is no vascularization, so I hope no. And in the gut, we are unsure, but we were able to detect them between PC week till PC week 11. So during the first trimester of pregnancy, they are.
They're there.
And
They're insulin positive. Sorry, I misspoke.
yeah, and what is quite intriguing to me is that in this paper from which is in this Nature Medicine from Egozi,
Hmm.
they are able to find them also during the second trimester of pregnancy.
Hmm.
So it could be the case that in the human adult gut, there might be some scattered insulin positive cells. We don't know. We don't know if they shut off or turn down insulin expression at some point.
Yeah, that's exactly what I was wondering about. It's like, are there any residual, you know, insulin positive cells or those cells that have the capacity to secrete insulin in the gut, you know, in the adult or in the childhood gut? It's just kind of curious. Well, we'll have to look deeper in the literature, I guess, for that answer, or maybe wait to see what you guys find out. Last call for questions. No questions. I think you did a great job of explaining the whole thing. That's probably why.
Thank
But
you.
yeah, feel free to reach out to Adrian or his laboratory. And I know it was great to hear that you're in close collaboration with Roberto Maloney. And because he's also, you know, just a star in the T1D research world. And it looks like you're getting a lot of clapping and accolades in the chat. Thank you. It was a great talk, interesting talk, great data. So congratulations, Adrian, and we hope to speak with you again soon in the future. Great, great talk.
Thank you very much, and thank you everyone for coming.
Have a great rest of your day. Bye-bye.
Bye.